Well, good evening. This is Miss Aber. You are enjoying a screencast, a videocast, whatever you would like to call it, a podcast if you have downloaded it and are listening to it on your iPod or other device. Tonight we are going to learn about the carbon compound known as proteins. And so you see a delicious assortment of proteins on your screen here. Proteins! Yay! Proteins. And this is more the biological depiction of proteins, and you can see that they are long chains folded up into a variety of ways. And we really think of proteins as these multi-purpose molecules because they have a variety of functions. They are also very structurally diverse, and structure and function, um, as we have discussed before, really are so key, and they're intertwined when we discuss proteins. The function of a protein, proteins are really involved in everything. They've got their hands in it all. So enzymes can be proteins. There are enzymes that are also nucleic acids. That's for another exciting topic. But enzymes such as pepsin, which are involved in digesting proteins, and DNA polymerase, which are involved in making long chains of DNA, are proteins. Structure, keratin and collagen, which are involved in the structure of the body. About a quarter of all proteins in your body actually are made out of our collagen. How about that? Pretty important. As carriers and for transport, the protein hemoglobin, if you have not heard of that, you will hear about it a lot. It is the protein that carries oxygen throughout the circulatory system, and it can bind oxygen and release oxygen when it needs to. And it's really quite important. Aquaporin is involved in transport. Aquaporins are the pores that allow water in and out of our cells. So also pretty important. Cell communication proteins are involved in as well. In signaling, insulin is a protein as well as other hormones, and the receptors on the cell that receive those communications are proteins. In the defense of organisms, those are proteins. Your antibodies are proteins. And in movement, actin and myosin, which are proteins in your muscle cells, those are proteins. And storage, the proteins inside bean seeds, well, guess what? I just said it. They're proteins. So really proteins, they're doing it all. So let's talk about the structure of proteins. The monomer of protein is an amino acid, if you can remember that from your freshman biology class. And so we have a picture of two amino acids here coming together to make a polypeptide chain. And as with all monomers coming together to make a polymer, they're going through the dehydration reaction, meaning that the hydroxyl group from one monomer and the hydrogen from the other monomer are going to be released to create a molecule of water. And then we have the peptide bond here between the amino group of one amino acid and the former carboxyl group, now simply a carbonyl group of the other amino acid. There are 20 different amino acids. You can see this R here. And the amino acid refers to how that amino acid can vary. As I had mentioned, the polymer of a protein is called a polypeptide. However, a polypeptide is not a protein. Okay, a polypeptide has to have shape. I'm sorry, a protein has to have shape. Whereas when we refer to a polypeptide, we're just talking about a polymer of amino acids. Okay, so this is very important. A lot of what we think about when we think of a protein is we think about its shape. And a polypeptide chain has no shape. It's just this linear chain of amino acids. Now, a protein, a final product of a protein, can be one or more polypeptide chains folded and bonded together. So you can see this in growth hormones, hemoglobin, are 4 different polypeptides binded together. So proteins really end up being these huge, these complex molecules. That tends to be the case. And really important to remember, the reason why we show these kinds of pictures when we're thinking about proteins rather than these kinds of pictures is because proteins are 3D. They don't exist flat on the screen, okay? They are 3D in shape and they interact with each other in 3D forms. And it's just so necessary that you know that about proteins. Rubisco is really quite interesting. It actually has 16 different polypeptides that come together. Rubisco is incredibly important to not just the plant world, but to all living things. Rubisco is the enzyme that's responsible for taking the carbon out of carbon dioxide in plants to get that whole Calvin cycle going to make sugar out of that carbon that it takes out of carbon dioxide. So if this protein is not functional, then no sugar is made in plants. Then we kind of go hungry, don't we? And that's no good. So let's talk about these amino acids. Structure. We always got to talk about that, don't we? So there's a central carbon in amino acids in which everything comes off of. And there it is, just like that, like magic. Sometimes I think that's what computers are. It's magic. Off of that central carbon, we have an amino group, ta-da, and then we have a carboxyl group, and ta-da. Okay, so see, knowing your functional groups is the cool thing to do. It is also the right thing, the morally upstanding thing to do. Yeah, the penguin gets it now. See, amino acid, amino group, carboxyl groups are acidic, acid, amino, acid, amino, acid. Okay. Also on every amino acid is this R group, or sometimes referred to as a side chain, and this varies based on which amino acid that you are peeping. Okay. And I've said all that. Yes. R groups also, for each different R group for the amino acid, it gives that amino acid a unique chemical property. And so you can think about it like there are, instead of 26 letters of an alphabet in the protein language, there are 20 different letters of the alphabet, 20 different amino acids. And you can make lots of different words. Proteins. So, the effect of different R groups. There are nonpolar amino acids, and here they are. So again, if we know that something is nonpolar, then we also know that it is hydrophobic, okay? You don't have those charges. You don't get along well with water because water is charged and it's polar. You just don't get along, right? So you can check out glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and old proline over here. Looking at their R groups are nonpolar, they're hydrophobic, and when these amino acids get into a protein, they will make either that entire protein or that part of the protein nonpolar and hydrophobic. We kind of answered that, didn't we? Why are these nonpolar and hydrophobic? Think about it. Look at the R groups. It's all about the R groups. So now looking at the polar amino acids, being polar means that you are also hydrophilic, right? Because now you have a charge, hydration shells can form around you, everybody's happy, they like each other, things are great. And so you can read for yourself here the different types of polar or even electrically charged amino acids. And see, you can even have acidic or basic amino acids, which would then, when these amino acids are present in the protein, would either make the entire protein, if there's enough of these amino acids, have that property, or that section of that protein be polar or acidic or basic. So, question, why are these polar and hydrophilic? Just look at the R group, right? The R group is polar, then the amino acid's polar. The R group is acidic, the amino acid's acidic. So basically what happens here is with the acidic side chains is it ionizes in water and it donates a hydrogen. And therefore, if they're a hydrogen donor, you increase hydrogen ion concentration in the solution. Therefore, you are an acid. These amino acids that have basic side chains, they ionize in water. And if I'm telling you that they're basic, so what do you think that they do to hydrogen ion concentration? They accept hydrogen ions, right? Yes, so they decrease the amount of hydrogen ions in the solution, and that's why we call them base. We could also call them hydrogen ion acceptors. And then there's some amino acids that contain sulfur. If you don't know this by now, sulfur stinks. That's just a good thing to know about life. If somebody says, hey man, I just bought some sulfur, it's really cool, you want some? Your answer is no, because it smells really bad. But the cool thing about amino acids containing sulfur is that they like to form these cool things called disulfide bridges. And yes, I do know that I've hit my mark as far as saying the word cool, so I will say it no more. No promises. Okay, so amino acids that contain sulfur form these things called disulfide bridges, and they are this interaction between sulfhydryl groups. So you have one sh and another sh in the Sistine Chapel, right? That's a joke for A block. You tell C block about it, A block. Okay, so there's a disulfide bridge between the two sulfhydryl groups, and it stabilizes this 3D structure. Okay, so say this amino acid and this amino acid see each other across the protein, and they make a link, a covalent link. That's pretty strong, isn't it? And why do you think perms smell like rotten eggs? It's because they're introducing these disulfide bridges. Haha, funny. Okay, how do we make proteins? We make proteins by building these peptide bonds. Peptide bonds are the bonds between amino acids. Specifically, they are covalent bonds between the amine, the amino group of one amino acid, and the carboxyl group of another. And we again do this dehydration reaction where the hydrogen leaves one and the hydroxyl group leaves another, and we remove a molecule of water. And we end up with a bond between carbon and nitrogen. We call this dehydration synthesis. So think about it. When you're dehydrated, you've lost a lot of water, right? Same here. They've lost a lot of water. Something to note that's pretty important is that polypeptide chains have direction. So we need to know this, because if you're looking at something that's one long chain, you want to know where the caboose is and where whatever the front end car of a train. Somebody tell me that tomorrow morning. Look that up, please. Okay, so polypeptide chains have a direction because you want to know where you started and where you ended, right? If we're both referring to the same protein and we said, I think something's weird with amino acid number 20. And I was like, no, I think something's weird with amino acid number 80. We could be talking about the very same amino acid. But what if, I'm just holding it upside down, which I probably am. We can fix that if we both say, well, how about if we both look at it from the same direction? And so there is an N-terminus and a C-terminus to polypeptide chains. So if you look at the backbone of a polypeptide chain, so you take the peptide bonds and you kind of straighten them out. Then you have where the amino starts, the amino group is. We call that the N-terminus. And then polypeptide backbone right here, amino acid, amino acid, amino acid. And then it ends, and where it ends, you should have a carboxyl group. And that's the C-terminus for the carbon that's ending there. And then the side chain should just poke out. at the top there. And then you should have this repeated sequence of NCC, NCC. N for the amine group, or amino group. C for that central carbon of the amino acid. And the other C for your carboxyl group. Then you have a peptide bond. And then it all goes over again. Get it? Huh? Anyone? Because if you get it, you're awesome. Polypeptides can also only grow in one direction, and we'll learn about this quite a bit more when we get to molecular genetics. So let's take a look at protein structure and function together. And really, really important is that the function of proteins depend highly on the structure. And in fact, if you mess up the structure of a protein, it no longer works. You mess with the structure, you mess with the function. They're like, proteins are like runway models. If you mess up the structure of their face, they can't work anymore because they're no longer pretty. I mean, they probably can get another job because, you know, they're people, but they won't be runway models in the traditional sense anymore. I hope I didn't offend anybody with that analogy. So anyhow, proteins have this 3D structure. As I had mentioned, they're twisted, they're folded, they're coiled into this unique shape that says, hey everybody, I'm hemoglobin. I'm here to pick up your oxygen, and then I'm going to take it to the cell and I'm going to release that oxygen. I'm so cool. Actually, really, kind of is cool. Okay, so if you can kind of imagine this picture of hemoglobin being 3D. Okay, same for pepsin. There's an active site right here where the protein will go move in and bind specifically with pepsin right there, and pepsin will munch on it and break it down. Think about collagen. If 25% of the proteins in your body are collagen, are you 3D or are you 2D? How about that? So how do we get to this long chain of amino acids, the polypeptide, to this 3D twisted, coiled, folded protein? Well, you go through 4 steps of structure in proteins. Your primary structure is the order of amino acids in a chain. Think of a series of beads together. And your amino acid sequence is determined by the gene that the protein is coded by, so the DNA. And you get something like this. So there's the N terminus, there's a C terminus, and it goes in this little chain. Any slight change in an amino acid sequence could affect the protein structure, and as I said, you mess with the structure, you mess with the function. So an example of this is in sickle cell anemia. So you have here some normal red blood cells. This is what they look like. These aren't donuts, they're red blood cells. And you have the normal structure of, this is the primary structure of normal hemoglobin right here. Now, if we change this 6th glutamic acid to valine, this is what happens right here. Just that one amino acid change. Because the glutamate is hydrophilic and the valine is hydrophobic. Wah, wah, wah. Sickle cell. Not good. But isn't that fascinating that just one amino acid change, one mutation, changes somebody from having normal red blood cells to having sickled red blood cells? Fascinating. So the secondary structure, this is what we'll refer to as local folding. This is folding along short sections of the polypeptide and adjacent amino acids. They're held together only through hydrogen bonds, so your weaker bonds based on the electronegativity of oxygen and that partial positive and partial negative charges. They form sections of 3D structure, either your alpha helix or your beta pleated sheet. So here you could have an alpha helix or a beta pleated sheet. And it's not like one protein is only alpha helix or one protein is only beta pleated sheet. A protein could have sections of both, but it usually is that there's a region of alpha helices and a region of beta pleated sheets. So here again, you can see that the alpha helix, it's a nicer picture of it, and the beta pleated sheet and the hydrogen bonding that's stabilizing it. The tertiary structure here is this whole molecule folding. So now the whole molecule is going to fold upon itself. And here's where you have interactions between distant amino acids. This is where you have hydrophobic interactions coming into play. Cytoplasm is aqueous, it is water-based. So any of your hydrophobic amino acids want to move away from this aqueous environment, and they're going to move away from that cytoplasm. Hydrogen bonds and ionic bonds also stabilize the tertiary structure. So here you have a hydrogen bond between two amino acids. You have ionic bonds here between two charged groups. Disulfide bridges are formed as well between the two sulfurs in your sulfhydryl groups in amino acids. And all of these interactions anchor this 3D shape of the tertiary structure. So again, in the tertiary structure, you no longer have this local folding, you have this whole molecule folding upon itself. The quaternary structure of the protein is when you have more than one polypeptide chain coming in and bonding together. So it's normally only here is when the polypeptide becomes a functional protein. And this is kept together through hydrophobic interactions. So again, here you have collagen, where you have 3 polypeptide chains winding together. Hemoglobin, where you have 4 chains coming together. A review here for your protein structure. Your primary structure is a chain. Secondary structure are local interactions, hydrogen bonding between the R groups, making either an alpha helix or a beta pleated sheet. Your tertiary structure is your whole molecule folding held together through hydrophobic interactions, ionic bonding, disulfide bridges, and hydrogen bonding based on amino acids. And then your quaternary structure is where you have your final protein held together through hydrophobic interactions. And that's where you have more than one polypeptide chain. You must know this. This is non-negotiable, absolutely necessary information. You need to know details about every level of structure, the bonds that hold them together, what's going on at each level. Okay. When you're talking about proteins, or when there's a question on the quiz about why did this happen and the protein's no longer functional, your buzzword is denature. You just say it. Warm-up question tomorrow is most likely the answer is going to be denature. So, and what denaturation is the unfolding of a protein. So as I've been stressing, structure means function for a protein. That if you unravel or untangle a protein and it no longer has the function that it meant to have, the protein no longer has the function that it meant to have. Okay, so think of it as you have a bowl and somebody came along and it's for some reason a very flexible bowl, and somebody came along and flattened out your bowl. Well, is it a bowl anymore? No, it is now a plate or a serving platter, but it's no longer a bowl, right? But if you had a spoon and somebody came along and poked holes in your spoon, is it a very effective spoon? No. So that's the deal with proteins. If you no longer have the function, you no longer have the structure. And that's what denaturing is all about. Denaturing is the unfolding of a protein. And any condition that disrupts hydrogen bonds, ionic bonds, or disulfide bridges will unfold a protein, will denature a protein. So things that can do that include temperature, pH, salinity, These all can unfold a protein, and you have done this through cooking. The most common one is temperature. You take a nice piece of chicken and you cook it and you eat it, and you have denatured protein. But if anybody has ever had ceviche, you have denatured protein through pH. Or if anybody has had like a salt cured meat, you have denatured protein through salinity. Denaturation alters the 3D shape of the protein. So you're altering the secondary and the tertiary structure. It destroys the functionality, but some proteins can return to their functional shape after denaturation. Many actually cannot, though. So that's cute. In biology, size doesn't matter, but shape matters and refers to proteins. So there you go. That's proteins. So how about it? So again, you're supposed to be taking notes, and that's what we're going to be checking out. Good night.